All right, let's get started. So at first, just wanted to call your attention to, maybe you got this email. In case you missed it, they're having a professional fair next Thursday. So if you're still kind of exploring what you want to do with your life, that might be something good to check out. And also remember, the next time we meet, we'll be having an exam. So I'm hopeful, I'm pretty sure, I'm like 99% sure that we're gonna get through 14 today. I will make that happen because I'm planning on the exam covering through process of infection that we started last time. Hopefully we'll finish it this time. So 4 different chapters. Make sure you don't forget about your homework. It's a lot of just kind of terminology and kind of reviewing stuff, not big, long, complicated processes this time, just kind of a lot of new stuff that you gotta review over and over again, maybe. So I do have office hours tomorrow afternoon. I will also be around Friday morning if you want to catch me, if you want to go over some things, and I'm glad to do that. Okay, so last time I wanted to write in the answers here. If you reviewed this, let me think about this. So number 3 is efflux pump. Number 1, alter the porin structure. That would be number 2. Let's see. This is number 5, multiplying. Okay, and number 4. So those were different mechanisms that we had of bacteria acquiring, or the mechanisms they have for antibiotic resistance. Just kind of understanding each of those mechanisms kind of helps to kind of draw out what that actually means sometimes. So if you have a chance to look at that, I thought that was a nice little exercise there. Okay. Otherwise, let's move on to where we left off last time. We were thinking about 3 different factors that affect pathogenicity and virulence. So remember, pathogenicity is really whether a microbe can cause disease, and then virulence is kind of a measure of the extent to that it's successful. So a highly virulent organism can easily establish an infection, and that infection is more likely to be severe in a host. So we looked at 3 different things. First, it depends on the species of organism. It depends on what kind of tools that it has in its tool chest that we're going to call virulence factors that enable it to be successful. It depends how many of those microbes the host has been exposed to, and how they're exposed certainly makes a difference as well. So just finishing up there, thinking about the strength of host defenses. So if 2 people are exposed to the same organism in the same numbers, 1 person could get ill and 1 person not get sick at all. So, you know, these things reflect very closely the factors that we went over that could make a bacteria that's part of your normal microbiome suddenly start causing disease. We talked about factors that could make an opportunist now suddenly cause infection. These are the same sorts of things. So there's just difference among individuals. You know, certainly the age of the individual, like we said before, very young or very old, they're more likely to become ill. Are they well nourished? Do they have any defects in their immunity, or do they have any, infections that would cause their immunity to be deficient? Are they under some kind of stress, either physical or mental? Have they had an organ transplant? They'd be on drugs that would depress their immunity. Are they, again, taking any other kinds of drugs? So all those things will affect. And in addition to that, as we'll see as we get into studying our immune system, there are just inherent differences in our immunity. One person can present certain kinds of antigen to our immune system that might make them more or less likely to get ill. So there are just genetic differences, even among healthy individuals. Okay. So now we're going to kind of follow the path that a pathogen might take when it causes an infection. So there are different, what we say, portals of entry. So there are different paths that a microbe can take in order to gain access to the host, to us. And some are more popular than others, okay? The skin is a potential portal of entry, but it's not a very common one because it turns out our skin is pretty good protection. And let me get back to the other ones in a minute. And I don't think I posted this. We're going to look at what our skin looks like in more detail next lecture. But just suffice it to say for now that our skin is not a common portal of entry because we have many layers of skin. The outside layers are dead. And so it's not easy for a microbe to attach to it. We have just, lots of mechanisms that makes the skin a pretty effective barrier. All right, so we'll look at that in more detail when we talk about kind of our lines of defense in immunity. As long as the skin is intact, we'll say, it's a pretty good barrier. The most common portal of entry by far is at our mucous membranes. Okay, so our mucous membranes, basically they line all the body cavities that are open to the outside. So places like the respiratory tract, our mouth, our nose, that's actually by far the most common portal of entry, the respiratory tract. We have defenses there, but they're maybe just not quite as good. And probably the second most common is the gastrointestinal tract, all right? So things we swallow. Urinary tract, that's a common one as well. Reproductive tract, the eyes, okay? Again, we have defenses, they're just not as robust. The placenta is not a very common portal of entry. There's only a few pathogens that have mechanisms to pass through that. And then basically kind of a catch-all for anything that's any portal of entry that's not, a placenta or mucous membrane is what we consider the parenteral route. Okay, that's kind of it's not parental, it's parenteral. So if you break that word up, this part of the word enteral, enteron is Greek for intestine. Okay, I guess it's a suffix, I don't know, or root of the word, I guess, will come up a few times as we move forward here. So that's intestinal, and then par means not or against. So if we say not intestinal, basically that means like not the normal route of exposure. So anything here, this is kind of a catch-all for not a normal route of exposure. Okay, so things like, oh, the skin gets punctured by an insect, for example, or a stab wound, or maybe a syringe. So some kind of, maybe you fall down, you scrape your knee. Okay, kind of like not the normal portals of entry there. That's considered a parenteral route. So once a pathogen gains access by one of those routes, and by the way, pathogens are usually very specific as to what portal of entry they're going to be successful at. So for instance, you can swallow HIV particles and you're not going to get AIDS. They can't attach to our gastrointestinal tract. You could breathe in HIV and you're not going to get AIDS. It has to be, specific portals of entry. Usually once that pathogen gains access to the correct portal of entry, the first step in establishing infection is going to be that pathogen has to adhere to a certain kind of cell. Okay, so we might call that adhesion or attachment phase. And again, that's usually the first thing that has to happen before that pathogen is going to be able to colonize the host. Okay, so pathogens are going to have special mechanisms in order to do that. Some examples, pili or fimbriae are specialized structures that bacteria might have in order to attach. For instance, E. coli has lots of fimbriae that help it attach to our large intestine. If it didn't have those specialized attachment structures, then it would just get washed out with our feces. In addition, organisms are going to have some kind of attachment proteins. And we usually refer to those either as ligands or adhesins. Okay, so even fimbriae, for example, will have attachment proteins at the end of the fimbriae. Okay, so proteins that will specifically fit some kind of receptor on the host cell. All right, so some examples. Again, here's a picture of fimbriae. Don't confuse that with flagella. Remember, flagella are for motility. Fimbriae are shorter. There's usually a lot of them, and they're going to have some kind of adhesion or attachment protein at the end of them. And then here's your Book's figures that illustrate that. So here's the bacterial cell. These little oddly shaped receptors, okay, those are the ligands. Again, sometimes they're called adhesins. I more often see them called ligands. And then here's one of our host cells. You can see the cytoplasmic membrane. And then here's some kind of receptor that the bacterial ligand would be specific for, so that it can attach. All right, and this on the right is just a scanning electron micrograph of some E. coli that are attached to our colon wall. This kind of shadow here, this grayish part here, that's showing a bacterial capsule, which can also help it adhere, but you can't really see the fimbriae there, but a capsule wouldn't be enough to anchor it as much as it typically would need to be. Another example, again, don't memorize the examples. I'm just trying to illustrate kind of the principle here. You've heard a lot, perhaps, about how we think COVID is, how it becomes successful and how it attaches to our cells. We have receptors, these ACE2 receptors. We have a ton of those in the alveoli of our lungs, so deep within our lungs. And it turns out those spike proteins that COVID has, which we could refer to as ligands, are specific for these ACE2 receptors. And the immediate thing that I ask is, well, why do we even have these ACE2 receptors? Well, they're important to cut certain proteins, and they help regulate blood pressure, among other things. So They perform a necessary function for us, and that's just what the COVID virus happens to be able to attach to. Another example, the bacterium that causes whooping cough, Bordetella pertussis, it produces, I think, 7 or 8 different protein adhesions when it infects our ciliated cells of our respiratory tract. Some of those are toxins that help it adhere. It's really important that that organism adheres to the outside of our cells because it never actually invades any further in the cell. So it has lots of different mechanisms to make sure that it attaches, so that the bacteria there in yellow, and you can see the cilia there. Okay, so again, attachment is important in order for infection to happen. All right, so most bacteria have other tools then, which we'll call virulence factors, that help them be successful at maybe further invading the host. Okay, so we're gonna talk about 3 different kinds of virulence factors for invasion: extracellular enzymes. Now you talked about some exoenzymes, same thing in lab. If you remember back a few weeks ago, in lab we talked about extracellular enzymes that break down macromolecules like starch or lipids in order for organisms to kind of get energy from that. These are extracellular enzymes that, again, are important for establishing an infection. So we'll give some examples there. We're going to talk about toxins, a couple different kinds of toxins, exotoxins and endotoxins. So we'll talk about the difference between those, as well as antiphagocytic factors. Okay, so we'll talk briefly about kind of steps of phagocytosis, and then some things that bacteria can do so that they are not engulfed by our phagocytic cells, our white blood cells. All right, so let's look at some examples of some extracellular enzymes. Okay, here's a couple of enzymes that are secreted outside of certain bacteria cells that target the epithelial cells of our body. So we have epithelial cells that line all of our that separate the inside from the outside of our body. And those epithelial cells are kind of stuck together by a glue that we call hyaluronic acid. So some bacteria can make hyaluronidase. Okay, that's an enzyme that breaks down that hyaluronic acid and helps them kind of invade and squeeze through those lines of epithelial cells. Another enzyme, collagenase. Okay, collagen is the main connective structural protein. It's the main structural protein in our bodies. So if an organism can break down that structural protein, all right, then we see here how it's invading deeper and deeper into tissue. So there's a couple examples, a couple more examples of extracellular enzymes. One, hopefully will look familiar if you've been to lab today already, or earlier this week, we talked about coagulase. So coagulase is an enzyme, again, that's secreted outside of the cell that can start the clotting process, okay, which is a complicated process, but the advantage to that for an organism, here's a bacteria that can secrete collagenase, can form kind of a clot around a cluster of bacterium. So it's kind of protecting itself from the host immunity. All right, so it's kind of walling itself off. So it gives it a chance to kind of grow to some critical number. And then eventually it will break out of that clot and release another extracellular enzyme to do that, kinase. And then it can kind of, again, grow in numbers and then until it's ready to kind of disperse and invade further. Okay, so we talked about in lab, our most pathogenic of our staph is the one that makes coagulates, and it's no surprise because that again aids its virulence, helps it to be more successful. Okay, so and there's different kinases. So the staphylococcus will make staphylokinase, the streptococcus can make streptokinase. So there's lots of different ones there. All right, toxins. It's very common for bacteria to make toxins. Viruses, no, they don't make toxins. I'm not sure if any fungi or protozoa make toxins, not that I'm aware of, but this is pretty much a bacterial thing. They bring about a lot of damage to host tissue. There's two types. Okay, we have talked about endotoxin already in the past. Okay, so remember, endotoxin is actually part of the gram-negative outer membrane. Okay, it's the same thing as lipid A. Remember, let me cut to a picture here just to remind you. I posted this a long time ago, so not right here again. But remember our structure back in chapter 3 of the gram-negative cell wall. Here's the cell membrane down here. We have a small layer of peptidoglycan, a thin layer. And then the outer membrane here is composed of a phospholipid membrane, as well as these blue structures here are the lipopolysaccharide. And lipopolysaccharide is just made of lipid A, as well as some polysaccharides that kind of hang out of the cell there. So lipid A, that is endotoxin. I know it's on the outside of the cell, so why do we call it endotoxin? It's just a misnomer. We used to think that it was actually produced inside of the cell. Okay, so endotoxin then is released by the bacterial cell to some extent when the cell is dividing. A little bit can be released, but especially when the cell dies. Released when the cell wall is disrupted, especially when the cell dies. Okay, so when a cell dies, that outer membrane, that cell wall starts to break down, and that's when we get a large amount of endotoxin released. So what problems does that cause us as a host if we have gram-negative bacteria that are all dying at once and releasing endotoxin? Well, we consider endotoxin to have low toxicity most of the time. There are some exceptions to that, but compared to exotoxins, fairly low toxicity. And it causes things in our body like kind of a low-grade fever, inflammation, and we're going to talk all about inflammation in another week or so and what that involves. A little maybe blood clotting, for instance, kind of, you know, malaise in the host, makes them feel really tired. Okay, so generally this isn't too big of a problem, but we do have to consider if you have a patient with a gram-negative infection, whether we want to kill all those gram-negative cells all at once. Because when you have them, you know, say you give them an antibiotic and kill them all at once, sometimes that inflammation that can result from the endotoxin might be bad. So sometimes you'll let a gram-negative infection kind of run its course. Sometimes you'll just say, hey, we have to add some anti-inflammatories when we treat this as well. The other kind of toxin, like I referred to, are exotoxins. Okay, these are secreted proteins. Okay, so where endotoxin is a lipid, exotoxins are proteins. So we just talked about extracellular enzymes, which obviously are proteins. How are those different than exotoxins? Extracellular enzymes are going to work really right next to the cell that secretes it, okay, very localized. Where an exotoxin, when it's secreted, it's going to work system-wide, so throughout the host. It has very high toxicity, and a little bit goes a long way. So it kills host cells even when it's very dilute at a low concentration. So let me write that in here. Secreted proteins that act systemically. So an example of an exotoxin is botulism toxin. I've read that a 6-ounce bottle of botulism toxin is enough to kill everyone on the planet. So when you say it works at low concentration, that sure does, okay? And in addition, the exotoxins are often very specific, okay? So they're gonna target a certain kind of cell most often. All right, so some examples of exotoxins. Sometimes we talk about exotoxins as in kind of a non-specific way. So for example, a cytotoxin would just be a toxin that kills host cells. There are neurotoxins. So these are toxins that target nerve cell function. Okay, botulism toxin happens to be a neurotoxin. It impedes the nerve transmission so that our muscles can't move eventually. Tetanus toxin, that's another neurotoxin. That works in a different way. It keeps your muscles from relaxing. So you get kind of the stiffening. Okay. Another kind of toxin, enterotoxin. So there's that root entero. Again, these are toxins that act on our cells lining our gastrointestinal tract. So a lot of foodborne illness is due to the production of cells' production of enterotoxin, making giving us diarrhea or causing vomiting. Okay, so here's your book's just kind of little comparison of endotoxin and exotoxins. Here's some of the results of lots of endotoxin release. And this table in your book, I thought was interesting just to kind of help understand the difference between endotoxin and exotoxin. Of course, only gram negatives are going to produce endotoxin because gram positives don't have that outer membrane. Again, exotoxins are proteins where endotoxins are lipids. I wouldn't really worry about the heat stability there. This antigenicity, so we're going to talk about that term soon. What is an antigen? An antigen is something that elicits an immune response. So exotoxins, yes, are really good. They elicit a strong immune response. And that's important because that means that we can use a toxin that's been altered a little bit in order to make a vaccine. Okay, so we have a tetanus vaccine. It's just tetanus toxin that's been deactivated. Okay, so we can teach our immune system to recognize that so we won't be harmed from it if we come in contact with it. An endotoxin has very weak antigenicity. So it's not possible for us to make a vaccine against endotoxin. At least we haven't figured that out yet. Okay, so and there's just some examples, again, of representative diseases, not to memorize, just sort of in case you're interested. Okay. All right, so the last kind of virulence factor that I want to talk about are antiphagocytic factors. Okay, so mechanisms to prevent that pathogen, that pathogen will have to keep it safe from our white blood cells. So let me review a little bit or present for the first time what phagocytosis is. Of course, phagocytes, these are white blood cells, and we're gonna talk much more about that next week as we get into our innate immunity. White blood cells that are going to engulf and remove invading pathogens. Okay, so for example, here we've got some white blood cell, say it's a macrophage, for instance, that's extending its pseudopods and engulfing some bacteria into a membrane-bound vesicle, which we refer to as a phagosome. Okay, and then to bring about killing of that bacteria, the lysosome, that organelle that holds a lot of digestive enzymes and some other enzymes that we'll talk about soon that will fuse with that phagosome. Now we call it a phagolysosome. And now those digestive enzymes, most of the time, are able to bring about the death of that pathogen. And it's just exocytosis here at the end, right? So most of the time, our phagocytes are successful there. But again, sometimes bacteria might have a strategy to either keep themselves from becoming phagocytized in the first place or to survive inside that phagosome. So here's just some examples. We've talked in the past about capsule production. So when a bacteria makes a capsule, that can help it to evade phagocytosis. Whether it's just, kind of slippery and those pseudopods can't get around it, or sometimes that capsule has a composition that our immune cells don't recognize as being not the same as self. Question. Would a capsule production be part of biofilm? So usually when cells are living in a mass of other cells in a biofilm, most often they do make a capsule to help them all stay together, yes. Okay, so again, our immune system has a way, like we find this a lot that, okay, we phagocytose, the bacteria has a strategy against that, and then we have another strategy against the capsule. So it's kind of an arms race. We have ways to deal with an encapsulated bacteria that we'll get to pretty soon, but that's one mechanism that can help the bacteria survive for at least a time. Some bacteria can produce leukocidins. That's a kind of cytotoxin that will form pores in the white blood cell. So it's like kind of the bacteria going on the offense and trying to kill that white blood cell. Another example is M protein. So this is a specific mechanism that an organism, Streptococcus pyogenes, has to evade being engulfed. So here's an example. This is Streptococcus pyogenes. On the left here, you can see these M proteins extending around the surface of the cell. And on the right, here's a Strep pyogenes that for some reason is mutant and can't make that M protein. And this, as to why this keeps the cell from being phagocytized, this gets ahead of ourselves a little bit, but we have certain proteins that we make called complement proteins that will bind to the outside of cells, and then the pseudopods of the phagocytic cells have a better chance of kind of grabbing hold of that cell and enveloping it. Well, if the cell has these M proteins, then our complement proteins don't stick. Okay, so that's a way to kind of evade a mechanism that we have to be more successful in phagocytizing cells. So we'll talk more about that later. Some organisms, they get ingested, but then they have ways to survive inside the phagocyte. And we won't get real specific here, but you can see here that here's a bacteria that's been phagocytized, and the lysosome is not able to fuse with the phagosome. So now we've got the bacteria just producing quite happily inside that phagocyte. Mycobacterium tuberculosis is really good at that. It actually lives and reproduces inside macrophages. It's like criminal hiding out in the jail or in the police station, maybe. Okay, so part of that is due to the mycobacterium's really strong acid-fast cell wall that helps it survive, but it has some other mechanisms as well. All right, so those are just some examples of virulence factors that organisms, mostly bacteria, have to survive. If we think for a minute now, what's going on, or how do we describe these stages of infection? We've been looking at kind of the pathogen standpoint. If we take a step back and think about, well, how do we describe the stages of infection in our body? Then if we look at over time, the number of microorganisms or the intensity of signs and symptoms. Usually signs and symptoms get worse as there are more and more pathogens within a host. And let me define that. Signs and symptoms, what's the difference between those two things? Signs are observable. So things like things a doctor could see. Rash, the patient is vomiting, the patient has a fever. Okay, we can easily kind of quantitate or see those things. When a patient has symptoms, these are things that are just felt. So maybe pain or malaise or headache. Okay, a patient says they have those things, but we can't really tell by looking at them necessarily. Okay. So when a pathogen enters some portal of entry of a host, okay, then initially there's always going to be at least a little bit of what we call an incubation period. Okay, so the patient doesn't have any signs or symptoms, but the pathogen is actively multiplying within the host. The length of each one of these periods is quite variable depending on what the pathogen is. For rabies, the incubation period can be months. For COVID, we think that is somewhere between 2 and 14 days, more like maybe 4 days or so. For the flu, it's more like 1 day. Okay, so that can be quite variable. The infections that we have a really hard time controlling are the ones where the host is able to transmit that infection even when they're still in the incubation phase. So that's one of the problems with chickenpox. People are very infectious even when they're in the incubation phase. And when they're in the incubation period, they're not showing any signs or symptoms. So they're out, doing whatever they normally do. Of course, this was the case with COVID as well. A problem, why we had trouble keeping it from spreading. People were infectious even in the incubation period. As that pathogen continues to multiply and kind of vague general symptoms develop, we call that the prodromal period. Okay, so that's when you wake up in the morning and your throat's a little scratchy and you feel like, oh, I think I'm coming down with something. Okay, you just feel not quite right. This is why for 2 years you've had to self-assess every morning, because we want you to pay attention to those kind of mild symptoms that you have that maybe normally you would just say, well, you know, maybe I'll feel better later. Okay, we want you to pay attention to those even more so now. And then again, as that pathogen reproduces even more, when you have the most severe signs and symptoms, that's called the illness phase. Sometimes at that point, a patient dies, okay? And that's the end. But assuming that our immune system starts to kind of eventually win out, then the signs and symptoms eventually kind of fade away. So we call that decline, and then convalescence when there are no longer any signs or symptoms. So that's how we label kind of the process of infectious disease in our bodies. And of course, for a pathogen to be successful, it needs to leave one host and go to another. Okay? It wants to keep spreading, keep making more of itself. So the ways from which it leaves one host and then either to go out into the environment, or we'll talk about where it goes after that, we call these portals of exit. Okay, so the one principle to understand is that usually pathogens are going to leave that host in much greater number than when they came in. All right, so you might have breathed in only a few measles viruses, but after that infection takes hold, you're probably coughing out, right, millions and millions and millions of more. Okay, so generally shed in large numbers. Those portals of exit, you'll notice that they mimic the portals of entry. They're the same ones. So respiratory and salivary portals, that's why we're wearing a mask right now. Hopefully for not longer, much longer, right? But that's supposed to catch some of those big droplets that come out when we speak or when we cough or sneeze. Fecal exit. So there are pathogens, right, that, you know, foodborne pathogens, for example, or when we drink contaminated water that cause increased motility of bowel. It's just a hard words to even say there, resulting in diarrhea. So let me switch to this picture. When you talk about motility of bowel, the first thing that comes up to my mind is cholera. And I don't know if you've read much about cholera, but it causes just a really devastating, super watery diarrhea that patients can have so much diarrhea that they dehydrate and die in as little as a day or two. So cholera patients, these are cholera cots, and they're so weak that they can't get up to go to the bathroom. So they just lay there and let the diarrhea drain out through them into the can. So the way to keep a cholera patient alive is to hydrate them. Hydrate, hydrate, hydrate. There's not, you know, antibiotics. I don't even think that they do a lot of good. You just got to keep patients rehydrated. So common in, you know, places where they don't treat their drinking water, especially like, you know, when there's lots of refugees, for example. Urogenital tract. This is less common. I mean, urine leaving in pathogen leaving in urine is less common, but of course, sexually transmitted infections will leave through semen or vaginal discharge. Bleeding, that's another one. Okay, so, you know, a blood draw or dirty needle, something like that. Okay, so those are your portals of exit. All right, so once a pathogen leaves a host, where does it go exactly if it doesn't go straight away into another person? Well, we call those sites where pathogens are maintained or living for, you know, short or even long periods of time. We call those sites reservoirs. Okay, and a reservoir can be either an animal, it can be another human, or it can be non-living. So when a reservoir is animal or human, typically that animal or human is not ill, to kind of fit the ideal definition of a reservoir. Of course, if it was just getting passed to another human and then they're getting sick, then we don't consider a reservoir. But humans that are not showing any symptoms and yet are contagious are considered a reservoir. Okay, so animals are really, really common reservoirs, not insects. Okay, we have a different name if an insect is involved here. We'll talk about that in a minute. So some kind of animal, like a bat, for example. Bats are reservoirs for a host of infections. There is good reason that most of us are kind of naturally afraid of bats. Lots of things, rabies is probably the first one that comes to mind, but bats live communally. They live like right on top of each other. And then during the night, they fly out and you know, travel long distances, maybe come in contact with other bats, and then they come back and, you know, roost right on top of each other and poop on top of each other and everything. So they spread infection around between each other. And when we come in contact with it, maybe through their droppings or, you know, some other way, then we can become ill. Okay, so infections that have animal reservoir, we refer to as a zoonosis. Zoonosis. Okay, or we might say a zoonotic infection. Okay, and sometimes infections that are kind of passed around among, say, animals, maybe mutate just a little bit, and then what we say spill over. So they those infections acquire the ability to infect humans as well. So obviously we think COVID-19 was a zoonotic infection. We think it probably has a reservoir in bats, and something changed just a little bit now to make it infectious to humans. So a lot of new zoonoses, you know, like Ebola, we think Ebola probably originated in bats, probably. A lot of them are particularly severe when they infect us. Okay, so another examples of a zoonotic infection is Lyme disease. So not the tick, tick's not the reservoir. We would call a tick a vector. We'll get to that in a minute. That white-footed mouse, as well as deer, are considered reservoirs for Lyme disease. Okay. So when we come in contact with them through a tick is when we can get the infection. Okay. So humans can be a reservoir. Like I said, we would also call them carriers. Oh, so we call them reservoirs, or when it's human, we often call them carriers. One example of an infection where humans can be carriers, meaning, again, the human's not ill themselves, but they can make other people sick, is typhoid fever. So there was an example of this they talk about in your book. The 1930s, I think a town was getting a new water pipe laid, and they had an outbreak of typhoid in the town. Like 40 or 50 people died, I think. And they traced it back to one of the men that was working to lay that new water pipe. He was habitually peeing in the pipe. And if that's not sticking it to the man, I don't know what is, but he would do that. And it turned out that he had typhoid fever. And then it's possible for your gallbladder to be infected. And even though you're recovered from the infection, you're shedding the bacterium in your urine and potentially getting other people sick. So other famous case, Typhoid Mary, she worked as a cook and was getting lots of people sick, even though she wasn't ill herself. Okay, so that's a good example of when humans can be a carrier. We can be carriers for other things like whooping cough. you can, have some of that bacterium just kind of loosely associated, not be ill yourself, but you can pass it on to somebody else. Okay, and non-living things can be considered reservoirs as well. Things like food or soil, as well as water. Okay, so foodborne illness, or whatever food that's contaminated that you ingest, that food can be considered a reservoir. Soil, not as many examples of that, but anthrax spores can be just kind of hanging out in soil for long periods of time. You can get sick. Or water, drinking contaminated water can be considered a reservoir as well. So how does that pathogen pass from that reservoir to humans? We talk about different modes of transmission. Okay, so we can have contact transmission. Okay, so a direct contact, you come in obviously direct contact with that reservoir. So it can be something like a handshake. It can be, kissing or sexual intercourse. Mother to baby would be considered some direct contact. Or you could have some indirect contact by a fomite. Okay, a fomite is a non-living object. So say you go in, you type your paper on the computer in the computer room upstairs. Maybe the person sitting there before you sneezed all over the keyboard. You touch the keyboard and then you rub your eyes and oh, now that keyboard is a fomite. Okay, you've gotten ill from somebody who was there 2 hours before you. Or the kid that plays with the toy, the ill child that plays with the toy in daycare in the morning, he goes home in the afternoon, a kid plays with that same toy and gets sick, right? That toy is then an indirect mode of transmission of some pathogen. Droplet transmission is then, sneezing, coughing, even talking. Those large droplets that are passed when you're close to someone, less than 1 meter away. Okay, so droplet transmission, we think this is the primary way that COVID-19 is passed. It has the potential to be transmitted other ways, airborne, but again, this is why we have a mask on right now. Okay, all right, another mode of transmission, vehicle, whoops, try that again. We can consider air a vehicle. Okay, so if you're further than 1 meter away, 3 feet, greater than 1 meter away, then that infection is considered airborne. So COVID has the potential to be transferred that way. We don't think it's common. Other things like tuberculosis, like measles, they can certainly be transferred long distances away. So it's carried on really tiny droplets, most likely, ones that stay suspended for a long time. Water and food can also be considered a vehicle. It depends how long the pathogen stays in that water or food as to whether we consider it a reservoir or a vehicle. If it's only there for a short period of time, then we would probably consider it a vehicle. Okay, and lastly, one other kind of transmission is referred to as vector transmission. Okay, so vectors are living organisms, usually insects. I actually couldn't find an example anywhere of when a vector was anything but an insect. So let's say vectors are insects that carry disease-causing microbes. Okay, and vectors can be either biological or mechanical. Okay, that's kind. I mean, they're both alive, right? So that's kind of the terminology is kind of weird there. But when it's considered a biological vector, then the microbe is growing. There's some part of its life cycle inside the insect. Okay, so think back when we talked about malaria, there's part of that plasmodium life cycle that takes place in the mosquito. Okay, so the mosquito in that case is going to be a biological vector. And most vectors are biological. If you think about like bubonic plague, there's part of its life cycle that takes place in fleas, and then we get bit by fleas. Vectors can also be mechanical vectors, and that just means the insect is a passive carrier. All right, so best example I could think of there is maybe a fly that lands on some manure in the farmyard, picks up maybe some bacteria from that manure, and then goes and lands on the potato salad at the picnic. Okay, and now the potato salad is contaminated. Okay, so it's just basically that pathogen is just kind of hitching a ride on the insect in that case. Okay, so we're going to finish up here. Oh, I just included this table just, again, not to memorize any of these examples, but it kind of gets you used to, using those terms, reservoir and different modes of transmission. So just sort of, if you're interested, that's kind of a nice chart. And I want to finish up with some terminology that we use in epidemiology. Obviously, a pretty hot topic right now. If you think you want to be an epidemiologist, it involves a lot of asking questions, taking surveys, asking people where they've been and what food they've eaten and who they've been around and what they've done. And also the statistical analysis of all that information. So certainly an interesting field, studying where and when diseases occur. So there's some terms that we use to describe the occurrence of disease, and those are incidence and prevalence. Okay, when we talk about the incidence of disease, that means the number of new cases in a given time. And then the prevalence is the total number of cases in that same given time. So here's an example. This is a chart that your book gives you. It's the incidence and prevalence of AIDS. My title must have got dropped off of that. This is a little bit of a confusing chart because the units for incidence here are on the left side and the units for prevalence are on the right side. And you can see the units are a little bit different. Okay, so you would never have the incidence, the number of new cases, be higher than the prevalence, the total number of cases. So that wouldn't make sense. They could be the same if a person died as soon as they got the disease. But you can see here the incidence of AIDS peaked in, say, 1993 or so at about 80,000. And at that time, the prevalence, if you follow, again, this over here was more like maybe 150,000. And as you can see over time, the new cases are definitely going down, but the prevalence has really skyrocketed. Can anybody think about why that might be the case? How is the prevalence so high if the number of new cases are going down? Good. Yes, people are living almost a normal lifespan with AIDS now. Okay, so they're living a lot longer. So even though we have fewer new cases, the prevalence is really high. Okay, so that just kind of gives you practice thinking about incidence versus prevalence. Some other terms that we use to describe disease, the occurrence of disease. Some diseases are considered sporadic, meaning you just have a kind of a case here or there. They don't seem to be connected to each other. Something like, I don't know, botulism, like, oh, we got one person that, you know, came down with that, you know, doesn't cause a big cluster of cases or anything here or there. An endemic infection is one that's constantly present. It might have some seasonal variation, for example, but something like the flu. We always have that. Flu can also be epidemic. So we get an unusually high numbers of flu at one time. And a pandemic is when we have that infection that's epidemic in more than one continent. Okay, so you should ask yourself, well, how do we know what's a usual number? There are lots of infections that if you go to the doctor and you get diagnosed with a certain disease, doctors have to report that to the CDC. So if you go to the CDC website here, and there's an example of this in your book as well. Hopefully, there we go. All right. I don't know how to get that to go away. Okay, so this is kind of small, but this is an example of the notifiable disease chart, and this was for week 9. So this was like 2 or 3 weeks ago, I think, that I brought this up. You can see the list of diseases here. Let's pick one that we've talked about here. We've got malaria. Okay, so you can look up and find out, whoops, malaria in all of the US. Oops, I guess I can't write on this web page. In all of the US, how many cases of malaria that we've had this week. And in the previous year, what's the week where we had the most numbers? There was one week that we had 51 cases in the US. So far this year, we've had 123 cases. And comparing that to last year, it's about the same. All right, so we kind of know what's going on. And you can look at it, the numbers by state. And so it's just a surveillance program, so we know, you know, to pay attention if, oh, suddenly we have, you know, a whole bunch of cases of measles, for example, you know, what can be done. Okay, so that's how we know whether to call something, you know, epidemic or not. And this chart, I'll come back to this in a second. So this chart just kind of, you know, graphically, you know, something that's endemic versus epidemic versus pandemic. So the question that's, of course, in the news lately is, when can we call something an epidemic? When can we change and call it endemic? All right, this isn't really a question that we've had to deal with because we haven't had like a new infection like COVID. But you know, it's kind of, now we're starting to think, well, COVID's always going to be with us probably. You know, so when can we consider it endemic instead of epidemic? And there was an interesting article about this in the New York Times a couple months ago that kind of addressed this issue. And it basically said when a disease is continually circulating in very predictable patterns, okay, according to the CDC. So again, we may have more of it in the winter and less of it in the summer, but we can kind of predict that, okay? So really, it doesn't just depend on absolute case numbers. It really has more to do with what level society is willing to tolerate. Okay. So what level of disease officials and individuals are willing to tolerate? So in other words, when we have a lack of social disruption due to COVID, when we go all about our business without doing anything special, then that's when we would consider it endemic. Okay, so you see the mask mandates dropping. You know, pretty soon they'll probably just consider it endemic. Okay. All right, so that is what I had for you about chapter 14. Do we have questions? Anything, questions, comments? I think I will go ahead and introduce our next just a little bit because I have a little introduction that I like to do about immunology. But this new immunology stuff, this will not be covered on Monday. So chapters 15 and 16, we get into our immune system, and I've often tried to think, how can I not spend quite so much time on this? But the problem is that it's so interrelated that when you talk about one thing, you kind of have to talk about the other thing too. Homework is a little bit more extensive here, a little bit more animations. If you haven't cracked your book yet this year, I would suggest that when we get into adaptive immunity, that would be a really good time to start. It just kind of helps you understand the whole picture when you read about it a little bit different way. So what is our immune system? Our immune system protects us from lots of things. There's lots of things assaulting us all the time that we don't even realize. Lots of different viruses and bacteria and fungi constantly trying to get access to our portals of entry. Our immune system also is really important, and I know I didn't post this picture, really important to repair wounds and to clean up dead tissue in our body. And unfortunately, it has the potential to kind of turn on us and act in inappropriate ways that can cause disease itself. Another picture that I didn't post because I didn't want to make you nervous, but immunologists are really fond of diagrams like this. It's super, super complicated, and it's kind of a young field, so we're learning more about it all the time. But the cells that we're going to talk about that act in our immunity, like our macrophages and our B cells and our T cells, their function is really interrelated. All right, they signal each other with cytokines, and don't worry, we're not going to memorize what all those cytokines are, but it's hard to learn about, say, what a B cell does with not understanding what T cells do and what macrophages do. All right, so again, it's all sort of interrelated, which makes it complicated to learn about. It's good for us though, because if we have some aspect of our immunity that maybe isn't working or functioning quite right, something else may be able to take over and share some of that. Okay, so the way we're going to approach this, so just a little overview, we're going to try to split it up into 3 lines of defense. Okay, so we'll start out with kind of our external physical barriers. So we'll look a little bit more closely at our skin and what's going on at our mucous membranes. We've talked about our normal microbiota a little bit. I want to talk about that in the sense of how that can help us in terms of keeping us pathogen free. And there's just some other kind of non-specific things. So this is all part of what we would call our innate immunity. That means these responses are going to be the same every time we're exposed to some pathogen. All right, so whatever pathogen assaults our skin, it pretty much, reacts the same way. It doesn't learn and react better the second time it's exposed to it. Okay, same thing with our second line. Okay, this is still considered innate as well, meaning the response doesn't really change from a second to a third to a fourth exposure. There's some exceptions to that, but that's kind of the general rule. So we'll look at the role of our white blood cells, things like complement proteins, how our cytokines work, the role of inflammation, the role of fever. Okay, and then lastly, our third line of defense, this is our getting into our adaptive immunity. That involves the work of B cells and T cells. Okay, our B cell reaction, we typically refer to that as humoral immunity. Humors are bodily fluids. B cells make antibodies that function in our blood. And then T cells, that's a cell-mediated response. There's lots of different cells that have to bring that about. Okay, so that's a little overview of our immune system. I want to pose a question to you. Just, you know, maybe that's all you know about the immune system. A little, would you rather? Okay, if you had to do without either your adaptive or innate immunity, which one do you think you would do better on if you didn't have? Okay, there is a correct answer here that's backed up by some research. So talk about that with somebody next to you just for a minute, and then I'll tell you which you should have picked. What do you think, Grace? What would you do with that? Okay, that's fine. So expect everybody to know. All right, so I hear some mumblings kind of each way. All right, so it turns out that you would likely live longer if you did without your adaptive immunity. So you want your innate because, and there's actually lower vertebrates only have innate immunity and don't have adaptive. So that might have helped you if you knew that. But interesting experiment, and this didn't I did post this and it's a little bit fuzzy there, but interesting experiment that backs that up. They can do crazy things with knockout mice. So they take mice and they've gotten rid of the genes that enable them to do certain things. So what they did is they took some SCID mice. So that's severe combined immunodeficient mice that do not have B cells or T cells, which is the cells in your adaptive immunity, but they do have polymorphonuclear leukocytes, which is some white blood cells that are phagocytic, and they do have macrophages. So they have some cells of the innate immunity, but not the ones of adaptive immunity. And you can see here would be the normal course of an infection, a normal mouse. You can see that innate immunity can kind of knock that infection back a little bit, but it can't quite get rid of it without the adaptive immunity. Where cells up here that do not have those cells of innate immunity, but do have B cells and T cells, that it just, the infection goes unbridled, goes its own way. Because it turns out that the adaptive immune cells, that the B cells and T cells need those macrophages and those PMNs to present antigen to them, right? So the adaptive immune cells, they really can't get kicked in without the innate immunity first. So I think it's a good thought experiment. It's certainly something interesting to look into. I think that's a good place to stop. And we will hope you have a good weekend. Enjoy the nice day. See you Monday.